with my mentors Lee Spanish and Adam Reed, who's my writing mentor. And we studied the characterization of well water, or the chemical and microbial characterization of well water in the Colorado range. And this is just a frame. This is more of a proof of concept project. It's not. Yeah. I'll just press it that way. Yeah. Okay. So here's just an image to help you familiarize yourself with my resource and just the area we're looking at. This image was taken from the City of Boulder Drinking Water Department. And as you can see, it sort of depicts the drinking water sources that we're relying on here. So the purpose of our study, there are a couple of purposes. One is to create a kind of a baseline data set for future studies. So we can, the project itself is not looking at any individual variables that may be affecting well water, but in the future we're hoping it can be utilized for such studies. And it will also just aid in our understanding of this resource that so many people rely on every day. So one thing I want to clarify before I move on is that this is a study using microbial ecology. A lot of times when people hear water and microbes, they think pathogens, but we're not really looking at that. We're looking at how what microbes can tell us about this aquatic habitat. Oh, and another reason well water itself is very significant is because it's more directly related to its surrounding environment. So we can use this information to study how the built environment and the outdoor environment interact. So this is just a little map of the locations I tested at. We have 6 site samples. So just want to clarify again, because we only have 6 samples thus far due to time constraints, we can't really make any conclusive findings with this data, but more look at the methods we initially used and what worked and what didn't work, and see how we can move on to be more effective and create this more comprehensive data set. So a lot of what I did was getting this project off the ground and finding volunteers and kind of publicizing what we were doing. And this is an example of one of the flyers we sent out to search for volunteers for this study. It was distributed by the City of Boulder Drinking Water Department. And before each sampling, we conducted a quick well owner survey just to get some background on how the owners themselves felt about their well water. Some of the things we looked at is household members, age of well, recent system flush. The reason this is particularly important is if they had like a Clorox flush, that would affect the data we were finding in the water. And any other treatment process, pipe material. And this is a 1 to 10 rating just on how they felt. Doesn't correlate directly with the data about their quality, taste, and odor rating. And then general concerns. So the sampling methods I used was methane sampling, DNA extraction, and cell count. And then the chlorine and the multimeter helped us identify some more general parameters. So we used the multi-probe initially before we gathered any water samples. So what we would use it for is we would run the water until the pH and the temperature stabilized, and that way we know we're directly drawing water from the well, not water. We're not testing water that was sitting in the pipe material, in the pipe and gathering material there. So that's just how we made sure that the water itself was what we wanted to be getting. And then once that's stabilized, we logged information on the probe, and it logged things like GPS, like location coordinates, pH. So once we ran the water and made sure that everything was stable, we conducted methane sampling. And so what's important with methane sampling is to make sure it's not exposed to air and it's kept airtight. So the way we did this is we ran the water straight into a tube, and once the tube was completely flushed of any air bubbles, we turned off the faucet and the valve at the end at the same time, so we had just a tube of airtight water. And then we ran it through this 2-gauge needle into a airtight gas-tight bottle. And so we used the gas chromatographer at the USGS lab to analyze our samples. Oh, one thing before I move on. One problem with this method was that the way we got the water into our bottles was we pushed on the tubing here, but unfortunately that created a vacuum and it depressurized slightly the bottles. So that could have skewed our data slightly, which we realized and we thought about, but we decided to analyze the data anyway because we didn't think it was significant enough to mess up our samples. And so the way we analyzed it was using this gas chromatographer. And just generally how it's used is we use a microsyringe to withdraw methane or the gas sample. And we injected it right into the ejector, which is heated, and it vaporized the sample. And the carrier gas, in our case helium, pushed it through these coils. And we use an FID, flame ionized detector, to analyze the methane. And this machine gave us this beautiful chart. And so this is an example of one of our most successful runs. We did encounter problems with the machine from leaky septum in the injector port. So we took that into account and made sure we did each sample multiple times. And so here's just a bar graph to kind of show the data we found for the methane samples. And as you can see, it's very up and down. So like these 3 sites have such little methane present, while these 3 have a lot more. So again, can't make many conclusions, but when we continue with this research and this study, if this haphazard pattern continues, it might be interesting to look at the environmental factors or human factors that may be causing this. The next method we used was we studied the DNA that was present in the water using DNA extraction. So what we use is Be Bright purged MSR bags to collect large samples of the water, and then we ran it through this funnel filter. One of the things we might correct for next time is we might use a cartridge filter instead. The cartridge filters are a little hard to use, but the benefit is that the water goes directly through the filter, whereas with a funnel filter, you have to open it, pour it, close it, open it, pour it, close it, which may cause some problems with keeping it completely isolated. So the filter collected the DNA, and then we conducted PCR. We extracted DNA and did PCR with it to replicate the DNA, and then we ran it through gel electrophoresis. And the way this works is it separates out the DNA segments into bands based on size. And as you can see, the different sites, at most of the sites we did negative control to make sure that the bags themselves were completely purged. And then we also conducted a negative control for the PCR itself. We also conducted cell counts to determine the cell load in the water. And so the way we did this is we collected conical tubes of formaldehyde, which pretty much stopped all cell activity, so you get a snapshot of the water at that particular moment. We then filtered it, and then stained the filters, or stained the cells, with rubidium iodide, which pretty much has a fluorescent property, and it intercalates in the DNA so that under a microscope we can count how many cells per field and use that to determine the cell load. And this is the chlorine meter, just to measure how much chlorine is in the water. It's pretty easy. Just get a vial of water right on site and measure it there. Nothing really significant with sound effects. And so, lots of future work. We were just as I was leaving, we created a partnership with Stephen Osborne, who's done a lot of chemical analysis of well water, and we were able to collect 40 additional samples. So it's going to be really interesting. My lab is going to be looking at the microbial side, and then his team will be focusing on a more in-depth chemical analysis. So with this, we'll be able to jumpstart this data set and have a lot more data to look at. So that'll be really interesting to see. And then this will help for future findings and an opportunity to study more specific variables. So special thanks to everyone involved in my project. Really appreciate all the help and questions.
Hi, Mia. Thanks for a great presentation. I was wondering, if you were to have more data, what sort of human or environmental aspects would you look at to explain some of the data that you got?
Yeah, so that's actually really interesting because originally when we started this project, kind of in the back of our minds, we were thinking something along the lines of Kayla's project about hydraulic fracturing, because currently what's going on in Colorado is pretty interesting. There's a moratorium on fracking in Boulder County, and then in Weld County, there's a whole lot of hydraulic fracturing going on. So hopefully, Kayla mentioned in her presentation about how she was using historical data and how each time they're going to build on this, they could look at new variables. So hopefully, we'll be able to contribute to studies like this.
Thank you.
I have a quick question. You don't have your data from your cell counts. You did the cell counts already?
Yeah.
This summer?
I didn't
comment on the variety that you saw.
It was actually really interesting because we took it from very different regions. As you can see here in the Google map, the samples were kind of spread out. So we had one sample that was in a burn area in the mountains, and so the cell counts, so the varying cell counts, some places had a whole bunch, like you could literally like see it, and we had to do several runs through the filter because it would get clogged so fast. I'm not really sure what causes that, if it has to do with depth of the well, but I didn't really include the data because I didn't think most of it was significant yet. So.
Thanks.
Did you know the depth of each well?
No, we don't. But we have the contact for all the well owners, and that's something we're talking about looking into more in depth.
Cool. All right. Thank me, I think.